very much. Sorry to get you back from coffee so quickly. I really need my 30 minutes, so I need to really get going on this. I've deliberately chosen a trendy title, really, just to provide a little short intro, very much to the organisers for inviting me to talk to you today. So the 4 phases of oxytocin, why did I choose this title? Mainly because Heroin Shrine Buddha, 4 phases in Bangkok, of course, which, interestingly, is the God of creation, mercy and benevolence, which perhaps you might think oxytocin is responsible for, although unfortunately it is not nearly as simple as that. Perhaps more importantly, it's a symbol of the fusion of different faiths. And that, at least in terms of oxytocin, represents the fact that economists, psychologists, neuroscientists, psychiatrists, philosophers are all hotly engaged in trying to understand the uses and functions of oxytocin in humans. However, for those of you who've been following the can be extremely confusing, with papers pretty much every week now on humans particularly, you might be forgiven for thinking that it's actually 33 phases of Buddha, which in fact the Heroin's Shrine was originally based on the 33 phases of Buddha. But I can assure you it's not really that complicated. As you would expect, oxytocin actually affects fundamental aspects of behavior, which of course ultimately will influence many different components of complex behavior. Just to remind you very quickly, oxytocin, the neuropeptide, was discovered, in fact, 107 years ago in the UK by Sir Henry Dale, and it means from the Greek "give birth." And it's a very small peptide, as you can see here, 9 amino acids. It has only 1 receptor. And most of what we known about oxytocin was that it was extremely important because for releasing the posterior pituitary to uterine contractions during birth, and also contraction of the myoepithelial cells in the breast for milk ejection. And here we have the classic axis here, release from the hypothalamus into the posterior pituitary, into the blood, and then affecting uterine contractions for myoepithelial cells in the breast. And the guys who are doing all of this, there are only a few thousand of them really, in the paraventricular nucleus and the supraoptic nucleus of the hypothalamus. Also another nucleus here as well. Are they interfering with one another? Okay. So these are the cells, but they also seem to have important effects on the brain as well as being released from the pituitary. And this was shown in a really quick summary of animal work. Larry, I'm sure, will be talking an awful lot more about his work and that of others. In fact, following on from Paul Pedersen's work in the United States, showing effects of oxytocin on maternal behavior in rodents, we first showed that it was released in the brain using a sheep model back in 1986 during birth and suckling. And the next year we showed that if you gave it directly into the brain of hormone-primed animals, you could stimulate both maternal care, but importantly, also the very selective bond between mother and infant that occurs in sheep within 30 seconds. Very, very powerful. About the same time, other groups in France particularly showed that it facilitates social recognition, particularly odours. And a huge amount of work has been done initially, Tsukada and Tom Insel particularly, but of course Larry Young has very much developed this field to a large extent. Voles, where it's found that oxytocin is released during sex and promotes pair bonds in social species, but not in non-social species. And this is reflected by a different receptor distribution in the 2 different kinds of species, particularly in brain reward areas. With the advent of genetic manipulations, there obviously have been produced oxytocin knockout and also oxytocin receptor knockout mice. And in general, a lot of studies have been done on it, but in general, shown that it's an anxiolytic and promote maternal aggression. It's important for social but not non-social recognition, and these social recognition effects seem to occur particularly via an action on the brain amygdala. The main pathways that are discussed, although there may be many more than this, are from the hypothalamus here to the amygdala that I've already discussed, also the brain reward areas such as the nucleus accumbens, and also the prefrontal cortex. However, you do have to kind of understand from the animal studies really what release of oxytocin within the brain is doing, okay? And often concentrations we found in animals, it seems also to be the case, in fact, in humans, are actually higher in CSF than they are in blood. In CSF, it has a much longer half-life than it does in blood, maybe 20 to 30 minutes. Many receptors are actually close to the brain ventricles, and so there may be a paracrine role of oxytocin being distributed within the cerebroventricular system. Also, oxytocin within the brain itself can potentially diffuse some distance from the site of release and not just affect the immediate postsynaptic site. So it can do a lot of things. It really is a hormone in many respects. So how does it influence behavior? And this is all really based on animal studies, and I'm going to go through it very quickly, but you need to understand some of the important principles about how this peptide, and in fact many other peptides, work. So it modulates its own release. It also potently modulates the release of a number of classical transmitters, noradrenaline, in fact also acetylcholine, dopamine, serotonin, and GABA. The behavioral effects reported in animals are usually associated with this modulation effect of classical transmitters. So the social recognition memory effects are blocked by alpha noradrenergic antagonists. The social bonding effects are prevented by dopamine D2 receptor antagonists. The anxiolytic effects of oxytocin in animals are prevented by 5-HT2A antagonists. A recent paper in Nature has also emphasized the social reward effects of oxytocin may be associated with 5-HT1 receptors. In the central amygdala, the effects of oxytocin are prevented by the GABA-A receptor antagonist, and also a paper has shown that it inhibits fear via amygdala GABA interneurons. And a final, very recent paper in Nature, which is very interesting, perhaps giving us a very great insight into how oxytocin may influence electrical activity in the brain, which shows that it appears to improve signal to noise. Signal to noise in neural circuits, in fact in the hippocampus, by enhancing fast spiking interneuron activity. So what about humans? Well, there have been a lot of release studies, and these are pretty much all based on blood concentration or saliva concentration measurements, and you do have to take all of these with some degree of caution, because they may or may not reflect changes going on in oxytocin concentrations within the brain. There are quite a number of studies showing that these two things can be sometimes dissociated. However, they also do agree on many occasions as well. So we have primarily, it's actually released as a consequence of sex, giving birth, suckling, and tactile stimulation, although in humans it's likely to be also released in response to a number of different social cues. It's correlated with things like strength of parent or mother-infant bonds, empathic responses to strangers, trustworthy behavior, and it tends to be reduced in individuals who have been in impoverished social environments. There's now a huge amount of work trying to identify, if you like, risk variants of the oxytocin receptor. I don't have time to go into them in any detail, but just to give you a flavor of some of the different polymorphisms that have been associated with aspects of social behavior. I won't go through them in detail, but you can see that a number of the polymorphisms are associated with key aspects of emotional and cognitive behavior, social behavior, including empathy and voluntary. And as you would expect, there has been huge interest, really, in possible therapeutic benefits of oxytocin for treatment of a wide variety of disorders. And in fact, many of these disorders have already been shown to be associated with various aspects of dysfunction in the oxytocin signaling system. At the end of the talk, I'm going to actually focus mostly on schizophrenia, and if there's time, I might also mention autism as well. Now, most of what we know in humans about the roles of oxytocin have been coming from studies where it's applied by intranasal route in order to get it into the brain. This is the only way that you can get it to the brain because it doesn't easily pass the blood-brain barrier. And so a huge number of studies have been using intranasal spray. The Novartis form tends to be the one that's used in the West, but in fact it's also available here in China. This is the Chinese version. Does it get into the brain? Well, in spite of the fact that everybody's been showing behavioral effects, it's only been in the last few weeks we actually published a study that proves, at least in humans, that it does increase oxytocin levels in the brain. Everyone was basing their assumptions on an original study by Bourne in Nature Neuroscience, which used vasopressin, which is a very closely related peptide, showing that it goes up in CSF and also in blood over fairly prolonged periods. They only went up to 80 minutes, though, along with other peptides as well. But they didn't actually look at oxytocin. And in a very small study recently, we've shown with patients who are undergoing CSF sampling for diagnostic reasons that you get a plasma increase in oxytocin. This is using an extracted assay, so it's about the most accurate way of measuring oxytocin you can get. It goes up relatively quickly and then it declines by about 75 minutes afterwards, whereas in CSF it actually doesn't go up significantly until about 75 minutes after application, which in general most studies don't start looking at behavioral effects until at least 45 minutes after intranasal application. Now, the four phases I'm going to talk about, which I think are relevant, particularly to psychiatric disorders, are the following. Its effects on attraction, bonding, and trust, and also a little bit in-group favoritism. Emotion recognition and empathy, because we've already heard people talk about empathy, I'm going to be very quick on this bit. Reduced anxiety and care and protection responses, and also, quite interestingly, work that we and others are doing showing relevance of oxytocin to influencing self-processing and also making of moral judgments. So there are already a huge number of studies in this area. I'm not going to go through each one individually because it will take too long. It increases attraction in men for neutral expression female faces. We've just also published about it in Psychoneuroimmunology. It's been shown by others. I'll talk a little bit about this. It's work that we've been publishing in the last years with René Hulleman and the University of Bonn. It increases the strength of monogamous bonds, and just last week, a paper that came out, we showed it increases the attractiveness of female partners relative to other women. It increases trust and also tolerance of broken trust, and it increases parochial altruism and also ethnocentrism. This is the Psychoneuroimmunology paper showing the very simple but well replicated effect that oxytocin increases the attractiveness of females to men. but we were interested in whether it does this via brain reward system and whether it actually does it via the dopamine D2 receptor. So this is a PET study. We find, yes, there's a nice increase in the activity in the nucleus accumbens, the reward area, related to the changes in interaction, but we find absolutely no effect whatsoever on changes in D2 receptor occupation. And this is raclopride PET. It's very sensitive. But we do find, however, changes in D2 receptor uptake in frontal cortex regions involved in attention. This is the paper that just came out last week. We did a really large-scale study. It's got a discovery and a replication component, just to make sure that everyone believes us. This is showing that behaviorally, men in a relationship, when they're given oxytocin, they show a positive bias in terms of rating attraction of their partner, but no effect on arousal, in fact. And this positive bias is for the partner, but is not shown towards unfamiliar or familiar other women. This is the degree that this happens is associated both oxytocin and placebo groups with the Eros scale, which is a scale of romantic. It's a romantic scale. What does it mean for brain reward areas? Well, as we would hope, it showed that we found increases in activation in two main brain reward areas, the accumbens and also the ventral tegmental area, only in response to the partner, not to either unfamiliar or in fact familiar women. So we do have good evidence for oxytocin affecting brain reward responses. Whether or not they involve dopamine is still an open question. This is another study, another aspect of social reward, which is the receipt of social touch. So this is men being touched by a woman, or when they think they're being touched by a man. In fact, they're always being touched by a woman, but they don't know that. And this showed that with oxytocin, it enhances the perceived pleasantness of the female touch, but not the perceived male touch. And there's an associated increase in the brain reward area, the orbitofrontal cortex. But importantly, in this study, both the behavioral and the neural effect were negatively correlated with autistic traits, so scores on the ASD. But these are normal, healthy subjects. These are not autistic patients. But of course, autism is a trait. This shows that we need to be cautious, perhaps, when we're going forward in using oxytocin for potential treatment of autism, because possibly there is a blunted response to oxytocin, both behaviorally and in the brain, in autistic individuals. Why? Well, this study, published a few years ago on autistic subjects by Gregory in DMC Medicine, showed epigenetic modification of the oxytocin receptor in autism, but importantly what they showed is that where you get increased methylation as a result of this, of the oxytocin receptor, you get a decreased expression of oxytocin receptor. This is actually in the temporal cortex of the brain. So it may be that there are epigenetic effects of the oxytocin receptor in autistic individuals which end up reducing the number of oxytocin receptors, and therefore it make it that much harder to stimulate behavioral changes therapeutically using oxytocin, although not in, possibly you have to use higher doses of oxytocin. So in conclusion, oxytocin may facilitate social attraction by increasing attention towards salient cues and making them more rewarding. It may promote the maintenance of romantic and other bonds via the brain reward system. In humans, the social reward effects may not only involve dopaminergic systems, and the two that are obvious as a potential candidates are opioids and serotonin, both of which oxytocin interacts with. Its ability to promote social reward may be blunted by autistic traits. Possibly this is a result of epigenetic mediated downregulation of the oxytocin receptor. In the second face, I'm going to go through this one very quickly because this is the one that most people know the most about. There's been a lot of work showing that oxytocin decreases amygdala responses to particularly negative emotion stimuli like fear faces. Also in relation particularly to autism, showing that it increases the ability to interpret emotions from the eyes or at the time looking at the eyes. Although importantly, a paper just came out showing that this is true for static pictures, but it's not actually true for dynamic ones, so the situation may be a bit more complicated. We also found recently that oxytocin increases emotional empathy, and another study has shown that it increases empathic accuracy. So generally at the moment from this area, we can conclude that oxytocin seems to decrease amygdala responses to emotional faces, but can also in fact improve face emotion recognition. This is mostly in terms of fear and anger. And although a number of studies have also shown it increases responses to or discrimination to happy faces, not all of them have. It's much more consistent with fear and anger, particularly anger. It increases the amount of time looking at the eyes in static but not dynamic face emotion stimuli, and it increases emotional empathy and empathic accuracy in some contexts. In terms of one of the much heralded aspects coming out of the animal work, which is the anxiolytic effects of oxytocin, this is becoming a lot more complicated in humans. Yes, the original studies showed that it decreases psychosocial stress, it reduces cortisol levels in men with early parental separation, increases positive communication during couple conflict, and enhances attachment security. But last year we showed that it also increases emotional modulation of startle and memory for aversive stimuli compared to neutral ones. This is a study using a classical startle task, acoustic startle task, which is known to be modulated by emotional pictures shown before the administration of the startle. In this case, negative emotional stimuli promote the startle normally, but whereas most people would have expected oxytocin as an anxiolytic to decrease this effect, instead what it did was to highly significantly increase it, showing that it's having a potential increase in anxiety and concern of potential risk. Furthermore, when we did a memory test for negative versus neutral emotional stimuli from the IAPS set, we found it didn't have an overall effect on memory, but it created a bias, oxytocin, for remembering negative pictures. These are mostly of victims. This is an example of some of the negative emotional pictures here. And the remembrance of these negative emotion pictures, was associated with an increased activity in the left insula that we've already heard mentioned by Jean in the first talk. So oxytocin, unfortunately, can potentially decrease, yes, but also increase anxiety, dependent upon the context in which you're giving it. It can actually make negative emotional stimuli more rather than less memorable, and this has potential implications for its use in, for example, post-traumatic stress disorder. It might actually make it worse rather than better. It may promote overall approach and protection rather than avoidance behaviors, which is really important, by facilitating insular responses, but correspondingly at the same time reducing amygdala responses. So you go towards things, people who are suffering, rather than run away from it. The final phase that I'm going to talk about is a far more complicated one, and obviously highly relevant specifically to humans. And a number of papers are now suggesting that oxytocin, given intranasally, can influence aspects of self-processing, positive self-perceptions, self-other face discrimination, It also influences activities, Chinese paper, in the default mode regions during self-referential processing. Also increases selfish behavior such as envy and gloating. Just to remind you why this might be very relevant in terms of psychiatric disorders, this is from a review we published earlier this year. This is a summary of the functional connectivity and activity changes. Blue is a decrease and red is an increase in the brains of schizophrenia patients in self-processing pathways in the cortical midline system in the default mode, and also in the mirror neuron system, particularly the insula. Notice only, I'm not going to go through this in detail, that there is a considerable reduction in connection between the front and the back of the cortical midline system, and also in activity in schizophrenia, associated obviously with fairly serious impairment in terms of self-disorder in schizophrenia. And this is fairly similar, severe in autism. And these two patterns are completely different, in fact the opposite to what you see, for example, in depression, where there's an increased activity in these self-processing. So if oxytocin can influence the system, it may have potential therapeutic benefit for treating self-disorders. And this is the first study that we've done, this is with René, showing that when individuals are asked to experience disgust, when they see pictures of disgust in terms of self as opposed to other, that there is oxytocin selectively increases activity in the cortical midline system, the anterior cingulate and the precuneus for self versus other, whereas in the anterior insula, which very much mirrors what Jean was saying earlier, you get an enhancement both for self and for other. This is we have a poster on this, Sarah will be presenting this tomorrow. This is a behavioral study in Chinese subjects, and it shows, in this case, using the endowment effect, which very simply is a measurement of self-behavior. It's the propensity that we have when we own things ourselves to actually want to pay less for buying them and get more when we sell them. Okay? And this shows that oxytocin enhances the endowment effect for self, but also, this is very important because self is not just you, it's an extended self which can include members of your family. Okay, so you also get effects with father and mother, but not with classmates in this case. So it's affecting not just self-processing, but also extended self-processing. This is moral decision-making, and this is quite interesting. We take moral dilemmas and split them into those benefiting self as opposed to not benefiting self, or they're not moral. And oxytocin only affects self-benefit moral dilemmas. It increases their acceptance. So basically it's like finding money on the floor and taking it for yourself rather than giving it back to the person who's lost it. Okay? It is increased in men, but it's actually decreased in women. So here is another example of gender differences in the effects of oxytocin, which have been shown now by a number of papers. So in conclusion from this section, we think that oxytocin does facilitate self-processing, particularly via the default mode network, anterior and posterior cingulate and precuneus, and also the insula. Oxytocin seems to promote selfish decision-making in men, but more altruistic behavior in women. It suggests that overall that in men it may promote more protection-orientated behaviors, supporting the family as well as themselves, possibly via the insula, but in women, more caregiving ones. And this may have potential therapeutic application for self-disorders in schizophrenia and also in autism. I'm going to go very quickly through what's been done with oxytocin in schizophrenia because I think it's quite encouraging. It's not a major effect, but studies are pretty consistent. In fact, oxytocin was first shown to have antipsychotic properties back in 1974. Many animal studies have actually demonstrated this using PPI and also phencyclidine treatment models. It's pre-pulse inhibition. Female patients with high oxytocin levels have less severe positive symptoms. And antipsychotic drugs actually tend to reduce CSF oxytocin. The oxytocin gene or polymorphisms of it are associated also with treatment responses. These are the large number now of either single shot dose or long-term treatment with oxytocin trials. And I won't go through them all, but basically they mostly show improved positive symptoms. Some of them also show some reduction in negative symptoms and improvements in aspects of emotion recognition and social cognition. This is the biggest one. It's actually carried out in Iran. It's an 8-week trial showing reduction in positive symptoms progressively over time compared to placebo. Importantly, there are no increased frequencies of adverse events. In autism, I'm sure Larry will also cover this in much more detail. The situation is still a little bit more mixed, to be honest. There are certainly evidence for reduction in repetitive behaviors, improved social interaction. These are all the single doses. Improved social skills. This was a 2-month course treatment. Also improved face emotion recognition, quality of life. Increased amygdala responses to faces and emotion recognition. The study here, which is the only one on young children, failed to find any effect of a 4-day treatment. And this one, just out in PNAS, shows increased activity in brain reward areas during the processing of socially meaningful stimuli. So it's encouraging, but it's still a bit of a mixed bag. And if you can see from that, there really aren't any big clinical studies yet published on autism. We have a lot of problems in this field, and particularly we need a much better understanding of dose responses and pharmacodynamics of treatments. We need to, very much in humans, establish the key neuromodulatory actions of oxytocin which influence behavior. It would be great if we could have oxytocin receptor ligands so we can image the receptor, but this may in the end prove to be difficult, if not impossible. Establish key polymorphisms and particularly epigenetic effects. Hopefully we might be able to identify some more centrally active agonists. And we need to try to understand better how to use it as an adjunct, particularly in the context of behavioral therapy and with existing drugs. It's not a miracle drug. Giving oxytocin by itself is unlikely to really promote major therapeutic changes. Also, there are many other areas where it might be potentially useful, which I won't talk to now, but including psychopathy. Pain certainly is an area that it's being shown to have advantages in. But most of all, we need more large-scale, well-controlled clinical studies in humans. And it remains just to say particularly thank you to a long-term collaboration that I've had with René Hullemann in Germany, in Bonn, and also to the members of my group in UESTC who have contributed to the study team. Thank you very much.